In lecture video B, we are going to be going through slides 13 through 24. Here we're going to be looking at how the environmental conditions that microbes are in out in the wild, out in nature, and then, in lab as well, are going to influence how quickly those cells can undergo microbial growth, how quickly they can divide. So when we start talking about optimal environmental conditions, it's important that you understand that these are a unique set of conditions that each microbe, each species, is going to have that is unique to their species. So there are some microbes that grow in very cold environments. There are some microbes that grow very well in hot environments. So each species is adapted to a specific set of parameters. Why is that? What is it that is making those cells capable of growing better in one set of conditions compared to another? Really, it comes down to how those conditions influence the stuff that's going on within our cells. We talked previously about how enzymes are a type of protein that are responsible for carrying out chemical reactions within a cell. So those enzymes are going to be influenced by environmental conditions. And if those enzymes are altered in a way where they can't perform their function or their reactions, either they can't perform it as quickly or they maybe are unable to perform their chemical reactions at all, well, that is going to affect, first of all, whether or not the cells can divide at all. It may end up killing the cells if we don't have those metabolic pathways that are needed, or we may see that growth is slowed down. So when we take those enzymes out of their needed range, then we are going to alter the three-dimensional shape of the enzyme, of that protein. Well, that's going to affect that three-dimensional active site. And if our substrate no longer binds to that active site, then we either can't catalyze the reaction at all, or if the active site doesn't quite match as well, then we will see that cellular growth is slowed down. So table 6.3 here is from your book. We're going to cover all of these different classifications, these categories that we can use for microbes based on the environments in which they prefer. Really what we're looking at is the environments that they function, that their enzymes function best in. So we're going to talk about all of these except pressure today. But this is a great review table for you to come back and kind of look through all of these terms at once. I'm not going to ask you for like specific temperature ranges or anything like that, but I expect that you have an idea of the kind of environment where we might find that classification of microbe. All right, so before we get going into this section, I would like to propose a question for you. So this image I pulled off of Google, but you can find several kind of depictions of this experiment where someone has gone out and purchased a McDonald's meal and they have let it sit out on a countertop indefinitely. So this one here, they let it sit out for 171 days. And over the course of that time, it doesn't really look like it's changed much at all. And it hasn't changed. We see no microbial growth. No decomposers are working to decompose this fast food meal. Now, the myth out there is that McDonald's meals don't rot because they aren't real food. I'm going to tell you that McDonald's meals don't rot. That is true. But they are real food. They are, the explanation for this lies in the conditions that microbes prefer to grow in. So as we're talking about this next set of material, I want to have kind of churning in the back of your mind, why do we not see microbial growth on this food? Okay? All right, so we're going to begin with temperature. We are concerned with environmental temperature that microbes are in because it's going to influence a number of different things. Remember that plasma membrane or that cell membrane is composed of a phospholipid bilayer. Well, membrane fluidity is going to be influenced by the temperature that the cells are in. That's going to affect how we get nutrients into the cell. Our DNA and our RNA molecules are not going to be as stable if we heat them up. Additionally, we talked about enzyme function. Remember, enzymes are three-dimensional proteins, and in order for them to catalyze their reaction, they need to have that active site the same three-dimensional shape as their substrate. So if we change temperature, we can affect all of these necessary things for microbes. What we can do is we can see that if we take them out of the range that they have evolved to be suited to, we can sometimes slow their growth. Usually, if we cool them down from their normal range, we'll slow their growth simply because we have made those physiological features not function nearly as well as they need to. If we heat them up even more, or heat them up like above their top limit of their range, we can see that those enzymes begin to denature. We completely lose nutrient transport as we're experiencing really hot conditions. So we have 4 different categories that we can have our microbes fall into. So notice that these are at degrees Celsius down here at the bottom, okay? So when we have 0 degrees Celsius, that's freezing. 100 degrees Celsius is the point at which water boils. So if we have microbes that have enzymes that are well adapted to very cold environments, where their membranes are adapted that way too, we call those psychrophiles. And they grow best at just slightly above the midpoint in their range. Mesophiles grow from about 12-ish degrees Celsius on up to about 45 degrees Celsius. Thermophiles like it nice and toasty warm from 40 to about 70. Our hyperthermophiles can grow even above temperatures that are beyond the point of boiling water. So when we start looking at these words, I want you to practice breaking them down. Phile means loving. So like mesophile is kind of that mid-range loving. Thermophiles like it hot. Hyperthermophiles like it really hot. Do you know what temperature the human body is in degrees Celsius? It's 98.6 Fahrenheit. Do you know what that is in Celsius? 37 degrees. So when we start looking at human pathogens, they are going to be the microbes that fall in human temperature range. That's the environment that we offer, that our bodies offer. So which of these 4 categories of microbes would we find most of our pathogens in? Most human pathogens are going to be mesophiles because their ideal growth rate is just at human body temperature. All right, we are going to talk just a little bit about osmosis so that we can understand the microbes that are capable of growing in what is called a hypertonic solution. So osmosis is covered in your textbook on page 113 and 114. So if you need to reference that part of the book, you're welcome to do so. But I really only want you to understand enough so that we can understand how some of our salt-tolerant microbes can grow in areas where most other microbes cannot. So osmosis, we said, it's the diffusion of water across a cell membrane. So when we look at diffusion of water, water is going to move based on its concentration. So how do we know the concentration of water? Well, we look to the substances that are dissolved within it. We call those substances solutes. So if we have a given water-based substance and it has a low solute concentration, say maybe 1% solute concentration, well, what does that mean for the water concentration then? Well, it would be 99%. If we had an area where there was like a 5% solute concentration, well, then it would be a 95% water concentration in that particular solution. So when we start looking at osmosis, we want to focus on the relative concentration of water across that membrane. Water is always going to move from its area of high concentration, which by default means that there's a low solute concentration, to an area of low water concentration, which by default means that it's going to be a relatively high solute concentration. So we have 3 different scenarios that we could have. First, let's look at what is called an isotonic environment. This is the easiest one. This is where we have the same solute concentration inside as we do outside. Well, if the solute concentration is the same, the water concentration is also the same. So in an isotonic environment, we're not going to see a net change in water movement. When we have a solution outside the cell that has high solute concentration, notice that there are a lot more red dots outside than there are inside. Well, that means that the water is in higher concentration here where the solute concentration is low. So in a hypertonic environment where the concentration of the solute is high outside the cell, osmosis is going to drive water out of the cell. A hypotonic environment is where we see a high solute concentration inside the cell, therefore a low water concentration relative to the outside. So in this instance, osmosis will drive water into the cell. I usually remember isotonic as the one that stands out because there's no change in our cells. Osmosis isn't causing water to move one direction over the other. So isotonic is where they're the same, no net movement. I remember hypotonic because of the O in hypo. This is when our cell is going to get big and swollen. Now, bacterial cells like a hypotonic solution because they have a cell wall that is acting as a cage and it's preventing the lysis or the bursting of the cells. Bacterial cells do not like a hypertonic environment because that water is going to be driven outside the cells and they're going to shrivel up. They're not going to have what they need. So let's watch a demonstration. This video, I like it because it uses eggs, just like normal chicken eggs that you would find in your kitchen. And they show the changes in size of the egg based on osmosis, the diffusion of water. Now, in this particular demonstration, they use sugar as their solute. We are going to be primarily concerned with salt as a solute, but the exact same principle happens, whether it's sugar or salt, but we are going to specifically focus on salt. So let's take a look at that video.
Today on Sci Guys, osmosis and naked eggs. Welcome to Sci Guys, I'm Adam.
And I'm Ryan.
Today we're going to be exploring osmosis through making naked eggs.
Egg-tastic! Egg salad! Excellent!
Close enough. Osmosis is the tendency of water to move across a membrane from a dilute solution into a concentrated solution.
The ingredients you need for this experiment are an egg,
Some vinegar,
corn syrup,
and food coloring.
and the equipment includes a glass.
And an old spoon or gravy ladle.
The reaction we're creating today isn't dangerous, but it's always a good idea to wear a lab coat or apron and goggles just in case of spills and splashes. The first step in our experiment is take your egg and gently place it in the bottom of your glass. If you break your egg, you're going to have to start over again. Place it in the bottom, and then pour vinegar over top of your egg. The vinegar is going to remove the shell off the egg and expose the membrane. The shelled egg will need to sit in the vinegar bath for 24 hours. As our egg bathes in the vinegar, the shell is slowly dissolved. The bubbles clinging to our egg will cause the egg to float, flip, and turn. After 24 hours, you will need to drain the vinegar and replace it with fresh vinegar to continue the process of dissolving the egg's shell. Let the egg sit in the vinegar for another 24 hours. After that, the shell will be fully dissolved and the membrane will be fully exposed. Once this step is complete, pour out your vinegar into a sink, gently catch your egg, and give it a rinse.
Once you have your egg removed from the vinegar, you'll notice it's grown in size compared to this raw egg. This is because water has flown from the vinegar into the egg through osmosis. But we're not done yet. The next step of our experiment is to place your naked egg into a glass, gently. Then, pour your corn syrup over top of it.
Bend your old spoon and use it to gently submerge your egg into the syrup. The naked egg needs to sit in the corn syrup for between 24 and 48 hours. The spoon is used to forcibly submerge the naked egg under the surface of the syrup because the egg will naturally want to float. If the egg floats, the surface of the exposed membrane may harden and it'll also cause this stage of our experiment to go much slower. Once this stage is complete, pour out your corn syrup using your hand to gently catch the egg and give it a rinse in the sink. As you can see, after a couple of days in our corn syrup, our egg is shriveled and small compared to the previous step where it absorbed a lot of the water from the vinegar. But we're not done yet. The next step is to fill a glass of water, add a few drops of food coloring, and give it a good stir. Now with your colored water, gently place your shriveled egg into the glass of water and leave it for a few more days. The shriveled egg will need to bathe in our colored water for about 24 hours. The egg will begin to grow and expand as the water passes through the membrane and into our egg. After 24 hours, pour out your water into the sink and gently catch your egg. Let's look at this experiment a little closer. First we need to look at why the shell of the egg is dissolved by the vinegar. Vinegar is made up of aqueous, or in other words, a water solution of acetic acid, and the eggshell is mostly made up of calcium carbonate. When we place our egg into the vinegar, the molecules in our glass cause a chemical reaction that produces carbon dioxide gas, water, and calcium acetate. Once the shell of our egg is removed and we have fully exposed the membrane under it, something really interesting happens. The water molecules in our vinegar begin to travel through the membrane of our egg and it begins to fill with water. The special thing about an egg's membrane is that it's semi-permeable. What this means is that it'll only let specific molecules pass through it. In this case, the molecules it lets through are water molecules. When we place our swollen egg into the corn syrup, the opposite effect happens. The water travels through the membrane and into the corn syrup. This movement of water occurs because there is an imbalance in molecules. The corn syrup has very few water molecules, but lots of sugar molecules. The egg has lots of water molecules and only a few sugars, proteins, and electrolytes. All the molecules want to be in equal proportions on either side of the membrane. But remember, only water is capable of moving through the membrane, so instead, the water moves from where there's a low amount of other molecules to where there's lots. This means that the water leaves the egg and goes into the syrup. As the water molecules leave our egg, it shrivels and shrinks. When we place our shriveled egg into the colored water, there's lots of water molecules, but nothing else outside our egg. Inside the egg, there is almost no water molecules and very few proteins, electrolytes, and sugar molecules. So once again, the water moves through the membrane to the inside of the egg, making the proportions of water to other molecules equal, which in this case causes the egg to swell. The process of water traveling back and forth through our membrane to create our different stages of eggs is known as osmosis. Well, that's it for osmosis. Thanks for watching.
Make sure you subscribe on YouTube, like us on Facebook, and follow us on Twitter.
And if you have any questions related to this experiment or about science in general, feel free to drop us.
There we go. All right, so like I said, that video showed you how they caused osmosis by changing the different concentrations of sugar, but the same thing happens with salt. So bacterial cells are fine in a hypotonic solution because they have that cell wall to prevent bursting. But it's this hypertonic environment that they don't tolerate well. So there are some species that have evolved mechanisms to counteract the movement of water due to a salt concentration that is high outside and therefore low water concentration. We call those species halophilic. So these guys are lovers of high salt concentrations, and they can grow, sometimes depending on the species, sometimes they can grow in environments that are up to 20% salt concentration. That's an insanely high salt concentration. Our primary genus that is halophilic is Staphylococcus. So Staphylococcus aureus, Staphylococcus epidermidis. Oftentimes, if we are trying to identify these salt-tolerant species, we will grow them on special salt-laced media. If we have an unknown species and we inoculate this salt-laced media with our species and it grows, it tells us that it's a halophilic species. And we can, with pretty good assumptions, assume that we're dealing with a staphylococcal species. So this should make sense to us. On our skin surface, we find staph living there as part of our normal flora. Same like as up in your nasal passages, in your underarms. These guys live in areas of the body that are relatively high in salt concentration, primarily because of sweat. So when we first started this section, we proposed the question, why doesn't a McDonald's meal rot? And the truth is, it is food, but we have done our best, McDonald's has done its best to try to make sure that no one gets sick. And so as part of their cooking processes, they create an environment that is just not hospitable for microbial growth. So the cooking of food is one of the ways in which we kill bacteria. And that's a good thing. If you're McDonald's, you want to make sure that you're not serving contaminated food. So this food is going to be cooked to a high temperature. The food, after it's been cooked, is going to be salted. Those delicious McDonald's fries are covered in salt. That burger patty has high amounts of salt as well. These high salt environments prevent microbial growth. Additionally, we see that the bread doesn't support growth primarily because it's going to dry out really quickly. But we also treat bread with a lot of preservatives to prevent mold growth. So most microbes that are going to affect humans are going to be neutralophiles. Now, your book uses the term neutralophile. They don't provide you with a figure, so I've pulled one from Google, and on it is labeled neutrophile. Same thing. So when we talk about our pathogens, they are going to like a relatively neutral pH in order to grow. The concentration of hydrogen ions and hydroxyl ions are going to play a role in cell physiology. Well, when we know that we're going to have these pathogens that like to grow in a certain range of pH, we can use that to, make it, again, less hospitable. So citric acid and sorbic acid are sometimes added to foods as preservatives. They are oftentimes in small amounts, but these acids help to create an environment that is less ideal for microbes to grow. That's why they preserve food. They make it less ideal for these particular microbes. So we do have some microbes, again, not human pathogens. We do have some microbes that prefer an acidic environment. We call those acidophiles, and the alkalophiles are those that like a more basic environment. But generally, most of our pathogens are going to be neutralophiles just because that's the environment that most of our body is going to provide. All right, we're now going to talk about the need for oxygen. It used to be thought that all living organisms need oxygen to survive. We now know that there are lots of microbes that don't need oxygen to survive. So why do we need oxygen? Well, if a microbe needs oxygen, the reason for that is because its chemical pathways are going to require oxygen at some step. So those organisms that undergo a pathway called aerobic respiration to produce the energy molecule ATP, well, they need that oxygen at the very end of their metabolic pathway of breaking down nutrient molecules. O2, oxygen, is what is called the terminal electron acceptor, meaning it's at the very end, but oxygen is going to be required to accept electrons and hydrogens. If we don't have that oxygen at the very last step, the whole pathway will shut down and we won't make ATP. Aerobic respiration of all of the possible pathways is going to lead to the most ATP production. So when we said that all organisms, or we used to think that all organisms needed oxygen, we have realized that that's not actually the case. In fact, for some microbes, the presence of oxygen can lead to the production of harmful substances. We group these molecules, these harmful substances, under the term reactive oxygen species. And we're just referring to a group of volatile oxygen-based molecules. So hydrogen peroxide is one example, but there are other reactive oxygen species that can harm microbial cells. So microbes, like I said, we thought we always needed oxygen. Turns out we don't necessarily. There are some alternatives that microbes have to aerobic respiration. That aerobic phrase means requiring oxygen. So anaerobic respiration is it's a metabolic pathway that is going to happen without oxygen. It does not require oxygen. We're still going to have a terminal electron acceptor to accept hydrogen ions and electrons, but whatever that terminal electron acceptor is, it will be something other than oxygen. So there's a couple of different options that we have, but anaerobic respiration does not require oxygen. Another alternative pathway that doesn't require oxygen is called fermentation. Again, we'll talk more in detail, but fermentation doesn't actually transport oxygen. It doesn't transport electrons. So we are not going to generate nearly as much ATP. In fact, we generate very little ATP from fermentation, but the benefit is that you don't need oxygen. So let's look at a couple of different classifications of microbes based on their oxygen requirements. If a microbial species requires oxygen because they are only going to be able to produce ATP through aerobic respiration, we call them a strict aerobe. This is the only metabolic pathway that they have to produce ATP, and they must have oxygen as the terminal electron acceptor, or they won't have enough ATP and the cells will die. On the other end of that spectrum, we can have what is called a strict anaerobe. These are microbes that cannot be exposed to oxygen or they will die. So they're going to make their ATP through another pathway that does not require oxygen. We talked about how sometimes oxygen can harm bacteria. Well, this is going to be one of those times where the presence of oxygen will lead to the spontaneous formation of reactive oxygen species. These are a class of molecules that are harmful to cellular components. And if you put a strict anaerobe in an oxygen-rich environment, reactive oxygen species will be formed, they will damage cell structures, and that cell will die. So if we have a species that is considered to be a strict anaerobe and we want to grow it in a lab, we have to create an anaerobic environment. And we do that using, what's depicted over here? This is an anaerobe jar, okay? So we've got our media in here. It has our strict anaerobe within it. In order to get that cell to divide and grow, we are going to use first of all, you have to seal the whole thing shut. And within that anaerobe jar, you're going to have a gas pack. So that gas pack envelope is going to be opened and it's going to work to generate some carbon dioxide, but most importantly, it's going to generate hydrogen gas within that chamber. Enclosed with that gas pack and your tubes of media with your microbe, we have a catalyst, and you can hardly see the catalyst in the lid up here at the top. But this catalyst is going to take that hydrogen gas that is being produced by the gas pack, and it's going to react with the oxygen gas that was trapped in that jar. It is going to result in the formation of water. So what you see when you do this is some condensation that forms on the sides of that chamber. That means when you see that condensation that we've had the oxygen removed. All right, so these are two possible options. Strict aerobes only perform aerobic respiration. Strict anaerobes will perform some type of anaerobic respiration or fermentation. These guys are going to be harmed by the presence of reactive oxygen species because they cannot get rid of those harmful compounds. So we have some I don't know, it's kind of a spectrum, I guess, of oxygen requirements. There are some microbes that are what we call facultative anaerobes, which means that they can live with or without oxygen because they have options in the different metabolic pathways they use to produce ATP. So our facultative anaerobes can perform aerobic respiration. They can also perform fermentation if oxygen isn't available. These guys also have enzymes that they produce within their cells that destroy any reactive oxygen species, those harmful oxygen compounds that are produced. We can talk about an additional kind of subset called aerotolerant anaerobes. These are anaerobes that can only ferment. They have enzymes, though, that destroy reactive oxygen species. That means that they can still grow in the presence of oxygen, but they can't utilize it to make ATP. We have a fifth classification called microaerophiles. Micro meaning small. They are lovers of a small amount of oxygen. They will only grow at low oxygen concentrations. These guys cannot ferment, so they require some oxygen in order to produce ATP. They have low levels of they have low levels of enzymes that destroy reactive oxygen species though. If they are in an environment that has too much oxygen, they simply can't keep up with breaking down those reactive oxygen species. Their enzymes, they just don't have enough. And so eventually, if there's too much oxygen, we'll have those harmful compounds made and it will destroy the cells. So how do we tell if we have a given species, like which of these categories it's going to fall in? Well, we can create what are called oxygen tolerance tubes, or sometimes called aerotolerance tubes, to look at where these guys are going to grow, and that can help us classify that species. So first we have to understand how aerotolerance tubes are made. So this diagram represents a tube that contains a liquid broth. That broth was heated up, and it in that process drove out all of the dissolved oxygen that was in that media. Additionally, we're going to add a very small amount of agar. What does agar do? Agar is going to result in the slight solidification of this media. It's not going to be as hard as what we might find in a petri dish, but it's going to solidify it just a little bit. So what happens is as this broth cools and begins to solidify just a little bit, the atmospheric oxygen that is contacting the surface of this media is going to begin to diffuse back down into the media. But the oxygen can only get into the media where the media contacts the air at the top. So what happens is that we get a gradient of oxygen concentrations within this particular tube. High oxygen concentration at the top, and we make our way down lower and lower and lower until there's no oxygen at the bottom. When we get this gradient established, we do what is called a stab inoculation of our culture. We take an inoculating needle, which is really just a wire that we've sterilized, and we introduce our bacteria by taking that inoculating needle and stabbing it all the way down and then pulling it straight back out. What that does is it introduces media all the way down, it introduces microbes all the way down the media, and it exposes those cells to different levels of concentration of oxygen all at the same time. And then we see where they grow. We can look at that growth to tell us information about the metabolic pathways that they're using, as well as whether or not they have reactive oxygen species destroying enzymes. So take a second. If you have printed off your notes, you can do this directly on your paper. If not, pause and sketch this out. I want you to draw dots on here where you think each of these different categories of microbes would grow. Make sure you take into consideration where the concentration of oxygen is going to be high, where it's going to be low, and where it will be absent. All right, have you done it? All right, let's look at where these guys are going to grow. Not just where they're going to grow, where they are not going to grow, and why they are going to die in those particular areas. Okay, so we need to first consider how much ATP can be made. Do you remember which of our metabolic pathways is going to give us the most ATP? Aerobic respiration is going to give us the most ATP. And then we're going to consider whether or not those harmful oxygen compounds can be broken down, because those could be lethal as well. So let's start with our strict aerobes. We sometimes call these obligate anaerobes, those names are synonymous. They're interchangeable. Most students get this one. If you're a strict aerobe, you need a lot of oxygen because this is how you're going to make your ATP. So all of the cells that were introduced into these lower oxygen concentration environments died because they didn't have sufficient ATP. Our obligate or strict anaerobes are only going to grow in an environment where they are able to metabolize and produce ATP in the absence of our oxygen. So when we come back, oh shoot, here we go. These guys we said are going to die in even the smallest amount of oxygen because the reactive oxygen species are going to be harmful to them. Well, these cells up here died because reactive oxygen species harmed their cellular components. Those oxygen compounds ended up killing the cells. So those are usually the two easiest. Now let's turn to the other ones. So facultative anaerobes can grow anywhere. We said that these guys can undergo aerobic respiration, but they can also ferment. So we see these guys, though, not growing evenly. We have more growth up here in an oxygen-rich environment. Why is that? These cells in the oxygen-rich environment are using aerobic respiration, so they can produce more ATP than the cells down here that are fermenting. Our aerotolerant anaerobes cannot perform aerobic respiration. So all of these guys are fermenting. Even in areas where there's oxygen that's present, they're not using it. So they're just kind of like slow and steady, chugging along, making a little bit of ATP. Now we have enzymes that destroy those harmful reactive oxygen compounds up here though. So they're able to survive. Our microaerophiles are going to grow just below the highest concentrations of oxygen. These cells down here that we introduced with our staph inoculation died because they lacked ATP. They can't ferment. These cells that were introduced and experienced that aerobic environment died because the reactive oxygen species destroying enzymes couldn't keep up with the formation of those harmful compounds. So understanding which environment we can grow in, which metabolic pathway we're using to make ATP, and then how we handle those harmful oxygen compounds, those reactive oxygen species. Can we break them down with our enzymes, or gosh, do we not have any enzymes? Are all going to play a role into how we categorize these microbes. All right, this is where we're going to stop for video B. We'll pick up with the next slide in video C.